Adaptation of the mitochondria, adaptations of the mitochondrion for its function. Now, I welcome you again to this next video after the video about mitochondrial structure, outer structure. Now, I want to see what enables this important organelle in our body that manufactures for us energy, what enables it to carry out its work. So, of course, the function is the main function we're talking about here, though there are many, but let's put forward the function of generating ATP. So, its function of generating ATP synthesis. How is it adapted to its function? But there are also other functions, as we shall see. But the main one is ATP synthesis, okay? That's very, very important. But you can also add on other functions, for example, lipid metabolism. Metabolism, you can also add it there. You can also add another function, for example, carbohydrate metabolism. Then we can also include another function, for example, amino acid metabolism. The mitochondrion can also be involved in such. However, the main function in this discussion here, first of all, is ATP, but we shall also hint on the other ones. How is it adapted for the functions it has? Now, we first of all credit part A of the mitochondrion, which is the outer membrane, which we said it has a protein called porine, making it porous. Porine makes it porous. So it allows passage of materials into and out of the mitochondrion. Which materials? For example, raw materials, which, for example, we can give an example of pyruvate. Pyruvate is an important raw material for the mitochondrion. It is the one which is metabolized to form the substrates. For example, to form acetyl coenzyme A, which is the only substrate for the Krebs cycle if it is to function towards ATP synthesis, towards the journey for ATP synthesis. We need acetyl coenzyme A. So pyruvate has to enter. But also after manufacturing ATP has to exit and go to the cell. So materials can cross to enter or to leave. Materials, for example, coenzymes, for example, reduced in NAD. Reduced in NAD has to cross the mitochondrion to enter. The one which is, remember, two molecules. From one molecule of glucose, two molecules of reduced NAD are made in the glycolytic pathway at reaction 6. So we expect reduced NAD there to be manufactured. So these materials has to enter, but also pyruvate has to enter. ATP being made has to cross and go to the entire cell. Plus other metabolites involved in various biosynthetic or catabolic reactions within the body. So that is very important. The outer membrane is for that. So that is the outer membrane. We said it is about 6 to 7 nanometers thick, which is a very good distance to allow the diffusion of materials to enter or leave the cell. Now, we go to part B. So it possesses part B. Part B is the intermembrane space, which we say that is about 10 to 20 nanometers in the wide, depending on the mitochondrion and the cell where the mitochondrion is functioning. Now, that one is very important. First of all, it creates a proton gradient. It has got a lot of protons there. So these protons are the ones that are involved in ATP synthesis as they are diffusing through the channel of the enzyme ATP synthase. So that energy they possess is transformed into kinetic energy, and in turn kinetic energy is coupled into ATP synthesis. So that is very important. It also contains proteins. It contains proteins that intermembrane space. It contains proteins, of which proteins, for example, the enzymes which are involved in aerobic respiration and also transportation materials to enter or leave the mitochondrion. Proteins are there in the intermembrane space. So that is also very important and an adaptation for the mitochondrion. It contains Part C. Part C, which is the inner membrane, inner mitochondrial membrane. This inner membrane, we said, it is about 75% proteins, meaning lipids are very few compared to proteins because there are also some other non-lipid, whatever, which may be there in the what in the mitochondrion. But 75% are proteins. What does it mean? Most of them are transporting proteins, others are enzymes. For example, proteins involved in the electron transport chain. So the inner membrane contains an electron transport chain, which is involved in ATP synthesis by oxidative phosphorylation. Okay, so that is very important. The electron transport chain is in the inner membrane there, which is involved in ATP synthesis. There are channel proteins which can allow entry and exit of materials. Remember, we said the inner membrane is selective in nature. It doesn't allow materials to easily go through just like the outer membrane. So that one is also an adaptation. Which adaptation? Possession of transporter proteins that allow selective passage of materials to enter the crystal of the mitochondrion, or and also possession of the proteins of the electron transport chain, which are involved in the movement of movement of protons from the crystal to the intermembrane space, but also possession of enzymes such as ATP synthase, the one you see, the one which is the ATP synthase, the one which is directly involved in ATP synthesis. That is very important. So we can also, on the inner mitochondrial membrane, we can also bring part D. Part D, which is the crystal for singular. This crystal, you see, it has got the stocked particles. Stocked particles, what does it mean? Crystal has got the ETC, electron transport chain, but also it contains the stocked particles. Those stocked particles are that is the ATP synthase. That is the ATP synthase you see there. It is having the F1, which is embedded, the F0, sorry, which is embedded in the inner mitochondrial membrane, and the F1, which is the head. This is ATP synthase that is involved in ATP synthesis. Always recall that ATP synthesis is not part of the electron transport chain. Okay, we shall see what constitutes the electron transport chain in more videos to come. That's very important to understand. Now, besides that, what we have to know is that one. Okay, now that is part D, but part D is part of part C, which is the inner mitochondrial membrane. That's very important. So that one also enables a mitochondrion to do its work. Now, a mitochondrion contains part E. Guess which part is that? Guess which part do you think is that part E? That is mitochondrial DNA. Mitochondrial DNA. Have you ever heard of cytoplasmic inheritance? This is part of it. So mitochondrial DNA, it contains genes. It is also governed by the genetic code. A code of 64 codons, as we shall see in some other biological discussions to come. So this genetic code is the constitution. It is the biological constitution. Biological constitution is the biological constitution, which is important. It is the biological constitution onto which cells use to synthesize proteins. So the mitochondrial DNA is involved in the synthesis of mitochondrial proteins. For example, these proteins may be enzymes, but also some structural proteins, like the ones that are to be embedded in the mitochondrial membranes, both the inner and outer membrane. Now, part F you see there, is the ribosome. That is ribosome. There are many. I've just given one. Is the ribosome. Now, this ribosome here, it is made up of the small subunit, the small subunit and the large subunit. The large subunit is 50S, Svedberg's constant. The small subunit is 30S, is Svedberg's constant, which are added to give us 70s, okay? To give us 70s, not 80s. So that is very important. What it is involved in what? It forms part of the biosynthetic machinery for protein synthesis in the mitochondrion. Which proteins are being made there? For example, protein Z, structural protein Z, and others, as we shall be seeing in other biological discussions to come. Now, there is also part G. This part G here, this part G you see here, that one is the matrix. This matrix here, the mitochondrial matrix here, it is semi-fluid. It is semi-fluid. It contains enzymes. It contains enzymes. It contains enzymes. Enzymes. It contains coenzymes such as NAD, both oxidized and reduced NAD. They are there. Reduced NAD, they are there. It contains ATP. It contains ADP. Then it contains the inorganic phosphates like that. They are found there. They are highly located there. Now, this matrix here, it is a site for this matrix here. You see here, it is site for the link reaction. Master. It links what? It links glycolysis to the Krebs cycle, because from glycolysis we get pyruvate, then pyruvate enters, but the Krebs cycle hates pyruvate like nothing. It can never allow to enter pyruvate. It has to first of all be shortened to 2-carbon compound, which is acetyl, then combined it with coenzyme A via a sulfide bridge to form acetyl coenzyme A, that is the substrate for the Krebs cycle or citric acid cycle or tricarboxylic acid cycle. Then the matrix also is the site for the Krebs cycle. You can also call it TCA, standing for tricarboxylic acid cycle or citric acid cycle if you like. So it is a site for whatever there. It is also a site for protein synthesis. When the mitochondrial DNA is coding for particular protein is required by the mitochondrion. Then it is also a site for lipid metabolism. They may be synthesized or they may be broken down to liberate energy from them. Also, they can also be oxidized. So it is a site for lipid metabolism. It is also a site for protein metabolism. Yes, site for metabolism. Some proteins can be metabolized in the mitochondrion. They can be metabolized in the mitochondrion there. So that's very important. Protein metabolism. So it forms part of. It plays a very big role when it comes to gluconeogenesis. Gluco meaning glucose, neo meaning near, genesis meaning beginning. Remember neonatal gluconeogenesis. So this gluconeogenesis is a process where someone's body is forming glucose from non-carbohydrate sources. They may be glucogenic amino acids, they may be whatever. So this one forms part of it. We may intend to reverse glycolysis from pyruvate. Instead of going to acetyl A, then we have to go back to glucose if need be. Then gluconeogenesis will occur. How does it unfold? We shall see how it occurs. So briefly, that is how the mitochondrion is adapted for its function. I remain Biology King Yugi. I urge you to follow my YouTube channel, Biology King Yugi, and also my TikTok channel, Biology King Yugi. But on TikTok, I'll be putting there some half videos. Then full videos will be there at YouTube. YouTube channel is still a young channel, but we need to grow it together, me and you. So feel free to comment, feel free to criticize, feel free to contribute, feel free to raise any concern. Be part of this video so that we transform the world biologically. Thank you very much.